In this lecture, we'll see about formation of urea. That is how the urea is formed from the nitrogenous waste material in the body of an organism. Under metabolism of amino acids, ammonia produced is toxic to the body. It is excreted out of the body as urea. This process of formation of urea occurs via the urea cycle for most terrestrial animals, and hence they are also known as ureotelic species. There are exceptions to this, like aquatic species which excrete ammonia directly into the surroundings, which gets diluted with the water, and they are called as ammonodelic species. Certain terrestrial species like birds and reptiles excrete ammonia as uric acid and hence are known as uricotelic species. The urea once produced is excreted out by the kidneys in the urine. Urea is also called carbamide and when dissolved in water has a neutral pH. Let's see about urea cycle. Ammonia is converted to urea in the hepatocytes or the liver cells in 5 steps via urea cycle. In the mitochondria, first two steps are there, and the last 3 steps that takes place in the cytosol of the liver cells. The urea then travels through the bloodstream to the kidney and is excreted in the urine. The urea cycle was discovered by Hans Krebs, who also discovered citric acid or Krebs cycle, and his student associate Kurt Hanselit in 1932. In the first step, there is the synthesis of carbamoyl phosphate which takes place in the mitochondria of the liver cells. Hence, here the ammonium ion react with carbon dioxide, which is the product of mitochondrial respiration, to form carbamoyl phosphate catalyzed by the enzyme carbamoyl phosphate synthetase first. This is an irreversible rate limiting ATP dependent reaction and consumes 2 ATP. In the second step, which is also takes place in the mitochondria, that is the synthesis of citrulline. Carbamoyl phosphate reacts with ornithine in the presence of ornithine transcarbamylase to synthesize citrulline. Just like oxaloacetate in Krebs cycle, ornithine plays a similar role, acting as accepting substrate at each turn of the cycle. Third step, that is seen in the cytosol of the liver cell, formation of arginosuccinate. In this ATP dependent step, the carbonyl carbon of citrulline is attacked by the lone pair of the amine in aspartate to produce arginosuccinate in presence of arginosuccinate synthetase. In this step, the second nitrogen of urea is incorporated by condensation. ATP is broken down into AMP and pyrophosphate. Next step in the cytosol, breakdown of arginosuccinate. Arginosuccinase promotes the cleavage of arginosuccinate to give arginine and fumarate in a reversible manner. Fumarate formed here joins the citric acid cycle, forming a link between urea and citric acid cycle. Formation of urea, which is the last step, arginine produced in the earlier step is broken down by arginase to give urea and ornithine. The urea formed here, it will be excreted through kidney and the ornithine is recycled back to the mitochondria for the next cycle. Thank you.